right, so to pick up where we left off, we are talking about nucleotides. They are organic molecules on the cell. They have carbon, hydrogen, oxygen, and they also have nitrogen and phosphorus too. But we were talking about the purpose of nucleic acids in the cell. And basically we talked about how DNA are the protein plans, the genes, which are plans for the protein. Or in the DNA, a copy is made, and the copy is called RNA, so it's the copy of the gene. And the cell will read and check out what the RNA says and turn that into a protein based on what the instructions in the RNA say to do. All right, I just want to be clear, the process of making a copy of the DNA, and the copy being RNA, this is called transcription. And the process by which the ribosome looks at what the RNA says, what the order of the nucleotides in the RNA says, and then uses that information to build a protein from it, it's called translation. And I love that these are literary terms, because to make a transcript, to transcribe something, means to copy something. It means to copy something down. You can ask for a transcript of someone's podcast sometimes, and it'll be a word for word copy of what everybody said. Whereas translation, it's like going from Spanish to English, or going from Greek to Urdu. I have a friend who is a professional translator, and she translates texts from, she's fluent in 7 different languages, and she translates between all 7 of them, going from one language to another. And translation is going from the language of DNA and RNA, which are made of nucleotides. I already drew this up there, but I can't spell nucleotides. Nucleotides into the language of proteins, which are made of amino acids. Nucleotides and amino acids are different things, and we will get into the process of transcription later in the semester, and we will go into all the nitty-gritty details of translation in a later chapter. Right now we got to look at what nucleotides are and what makes up a nucleotide. And I'm doing it again. I'm doing this vertically. This looks weird. Okay. All right, so we got this. So both DNA and RNA are polymers, meaning they're really big molecules made up of a bunch of smaller molecules all linked together. So nucleic acids, DNA, and RNA are the polymers. The subunit, or the monomer, is a nucleotide. And a nucleotide is made up of 3 parts: a sugar, a nitrogenous base, and a phosphate molecule. So let's just write that down. A nucleotide equals a phosphate molecule, and I'm just going to put a P with a circle around it. That stands for phosphate, or this whole PO4, sometimes -3 thing. It's a mess to draw. It looks kind of like, well, if I'm going to do phosphorus, I guess I should do yellow. It's usually, attached to the rest of a molecule over here. Right there. Who wants to draw that? No one wants to draw that. So what we do is we just do the letter P with a circle around it, and that's sort of an abbreviation for this, hot mess over here. So a nucleotide is a phosphate group, like that, plus a nitrogenous base. Nitrogenous meaning it has nitrogen. Base, it actually acts as to be basic. And plus a 5-carbon sugar. Sometimes it's called a pentose sugar. Pentose refers to just there being 5 carbons there. So let's take a better look at a better drawing of this. Here we go. Right here, and if I take a screenshot, I can annotate it a little bit. Yeah, we'll keep it red. Okay, so here's our phosphate group right here. Oh, look, this one they're leaving the the hydrogens on right there. So our phosphate group, our 5-carbon sugar, and they're telling us this is deoxyribose because there is no oxygen right there. If there were an oxygen right there, that would be ribose. And then a nitrogenous base, this one happens to be adenine. Okay, so nitrogenous base, meaning it's got nitrogen in it right here. All those nitrogen atoms. And remember, the things at the corners here, those are carbon atoms right there. Just because we don't draw them, we just have to know that they are there because this is organic chemistry and we just come to an agreement as organic chemists that we just don't feel like drawing all those C's. So if we don't put a letter where a corner is, we just assume it's a carbon. And this whole big old thing up here together, that is an adenine. If it looks familiar to you, it actually is part of the ATP molecule that I showed you in the last lecture. Very similar. So a nucleotide is actually very similar to the ATP molecule. That's so cool. Righty. No, I do not want to save that screenshot. Okay, so every nucleotide is made up of a gazillion of these linked together. I just want to say that this base here that's highlighted... How did I do that? What do you know? I'm in a different app. I'm not in my Photos app here. Okay, so this nitrogenous base could be several different things, but in this case, it's adenine. Let's look at how we take a whole bunch of these 3-part nucleotides and put them together. So how are the nucleotide subunits joined to make the polymer? Here we go. So I'm going to need another screenshot for this. So this right here is a whole nucleotide, right? I'm going to erase that, but just so that you know that this is what we're looking at right here. This whole nucleotide. Oh, and then down here, this is a whole nucleotide. So we're joining these two together. Alrighty, so hanging off this sugar here is an OH group. You can see that there. Okay, this is deoxyribose, otherwise there would be an O written down here, and there's not. All right, so an enzyme comes along and is like, I gotta get together this OH group with this hydrogen from this other nucleotide that's down here. Rips them out as a water molecule, and this is a dehydration reaction. RXN is just my little way of abbreviating reaction so that I don't have to write a whole big long word out of that. Oh my gosh, you all. My dog is in the room with me here and I can hear her snoring. I don't know if you can, but if you're wondering what that weird sound is, that is my dog snoring. She's an old dog. But like, if you can hear this, it's pretty hilarious. I don't want you going, what is that in the background? It's my dog. Okay, so a dehydration reaction puts together 2 nucleotides. So now we have this nucleotide here and this nucleotide here joined by the covalent bond. Notice that it is a sugar molecule here that is joined on one nucleotide that is joined to the phosphate group on the next nucleotide down. Okay, so this is a mess, isn't it? There we go. Meanwhile, this base up here, I don't know, maybe that's adenine. And this base down here, I don't know, maybe it's guanine. They're not connected to each other. There's no bond linking them together like that. Nope. They're only joined from the sugar of one nucleotide to the phosphate group of the next one down. Oops, I'm still on eraser. So what you get is over here on the right, we're looking over here, you've got a phosphate group, a sugar, a phosphate, a sugar, a phosphate, a sugar, a phosphate, a sugar, a sugar, a phosphate, a sugar, a sugar, phosphate, sugar, phosphate, on and on and on and on and on. So we actually call this thing a sugar phosphate backbone. Right there. This is the sugar phosphate backbone, this part here. I'm going to erase that now. Right. And meanwhile, all these bases here are hanging off the sugar phosphate backbone like teeth on a comb. Like if you go to comb your hair, it's going to look, you know, like the comb is like that. It's like this is the sugar phosphate backbone and all these teeth are the bases hanging off of it. Meanwhile, this whole thing is called a strand. So this would be a strand of DNA right here. So this part is the sugar phosphate backbone, and this whole thing, this hot mess that I'm making here, is a strand of DNA right there. Okay. So a dehydration reaction puts together two monosaccharides to form a disaccharide or a polysaccharide, and a dehydration reaction puts together two nucleotides to start forming a long strand of DNA. All right, now you're seeing these G's, A's, G's and T's and C's things over here as the bases. We need to talk about the differences between RNA and DNA. So let's do that. Okay, I'm not really sure why they put these lines in here in Notability, but DNA and RNA are very similar, but they have some differences in their nucleotides. The first up is that the sugar in DNA is deoxyribose. So we've seen that the picture of that, it's ribose that's been deoxified. Ribonucleic acid, RNA, the sugar, is ribose. So they're really close, but not quite the same thing. They also have different bases. The nitrogenous bases in DNA are guanine, or just the letter G, adenine, or just the letter A, cytosine, or just the letter C. And the last one, you probably know, it's thymine, and can you see it coming? Just the letter T. I'm not ever going to write these words out again. We're just going to use G, A, C, and T for them. RNA has most of the same bases. They have guanine, adenine, cytosine, but they don't have thymine. It's uracil. We say in RNA, U replaces T. You are not going to find thymine in RNA, and you will not find uracil in DNA. You will not find ribose in DNA, but you won't find deoxyribose in RNA. This is so that the cell can tell the difference between the original and the copy. If you think about the telephone game, you've heard of the telephone game before probably. You know, if you have a group of people and one person says one sentence and passes it on to one person, and the next person passes it on to the person next to them, by the time it makes it all the way back to the first person, it's a completely different sentence, right? Because when you copy something down 1,000,000 times, if you make a copy from a copy and then make that copy from a copy and then make the next copy from another copy, and oh my gosh, I'm going crazy here, right? You need to occasionally go back to the original just to remind yourself, wait, what did the original say? Make sure we haven't been paraphrasing that. Likewise, the cells often needs to go back to the original to see what the original protein plans say, because mistakes can get made when copies are made. And one of the ways that the cell does that is that it can sense the difference between deoxyribose and ribose, and it knows when there's a U in something instead of a T. So that's one way. And the cell really does know how to tell the difference. I can tell you this right now. DNA, as you know from like things like DNA fingerprinting, DNA hangs out on a surface for a long time. RNA is not very stable. It gets degraded after several hours. It'll get degraded, meaning chopped up. So DNA is so stable. RNA is not. There are enzymes called RNases that are especially prevalent on human skin. So if you're working with RNA in the laboratory, you have to be really careful. You always have to be wearing gloves. You cannot be touching even the tubes or the pipettes with your hands, because RNase is going to just chop up that RNA. So RNA is not very stable, because those enzymes are very specific for anything with ribose as opposed to deoxyribose in it, and uracil as opposed to thymine in it. Okay, the other difference between DNA and RNA is that DNA is double-stranded, whereas RNA is single-stranded. So 2 strands make up DNA, 1 strand makes up RNA. Now, to show you a little bit about the strands of DNA, I want to go back to that photo here. So if we're looking at the strand on the right-hand side here, it'll just be easier if I take another screenshot. Notice way up here on the right, I don't want to use that marker. I'd rather use the red pen. Right up here, notice there's a 5' written there with a P. Okay, that's the phosphate end of things. If you look at the middle here, over here, there's a phosphate group hanging out at the top of this one nucleotide, and down here there's the sugar end down here. So if you keep stringing a whole bunch of these, like if we put another phosphate group and with another sugar and another base, I'll just write the word base here. If we connect those there like that, there's still going to be an OH group hanging off down here. And if you look over here on the right, there's an OH group down here, and it says 3' end and 5' end. I'm pointing this out because the 2 ends are not the same. You've got a phosphate hanging off one end, and you've got the OH group hanging off on the sugar down here. And if we put a whole other nucleotide onto that, somebody's like, well, can't you just put a phosphate group on the end of that? Right there, can't we just put a phosphate group there? And the answer is no, because that phosphate group comes with a sugar and another base right there. So you're going to end up with another 3' OH group there. So I'm pointing this out because the 2 ends are not the same. You have a 5' end and you have a 3' end. They are not the same. And FYI, if anybody is like, why the heck are they calling that phosphate end the 5' end? And why the heck are they calling that sugar OH end the 3' end? What is this 3 and 5' thing? Why didn't they just go with 1 and 2? Because that's where numbers start off, right? And the answer to that is, look over in the middle here. Can you see this tiny little number 1 right there in the middle? I'm looking over here, folks. This is the first carbon. This is carbon number 1 on the sugar. This is carbon number 2, carbon number 3, carbon number 4. This is carbon number 5. The phosphate group is hanging off of the 5th carbon, so that's why it's called the 5' end. Whereas the OH end, here's carbon number 1, carbon number 2, carbon number 3. The OH group is hanging off of the 3rd carbon. So that's what we got there. And that's why it's 3 and 5, not 1 and 2. I didn't make this stuff up. I just spit it out for you. All right. So when Rosalind Franklin, she was actually the one who really figured out how the 2 strands of DNA worked out together. She didn't get, well, she got some credit for it, but she actually died before the Nobel Prize was awarded, and she surely would have won it along with James Watson and Francis Crick back in, 1954, when they won the Nobel Prize, along with someone else for figuring out the structure of DNA. She had died. Rosalind Franklin had died of, I want to say, ovarian cancer before then, and the Nobel Prize is not awarded posthumously, meaning it is not awarded to dead people. And it's really too bad because she got lost in history in that. She was a British scientist who did, she basically took X-ray pictures of DNA. And she was the one that figured out, the 2 strands of DNA, like this strand right here, and we need a different color, and this strand right here are held together so that the 5' end on one strand is opposite the 3' end on another strand, which means down here that where there's a 3' end down in the left-hand corner, over here opposite it is a 5' end. And notice that this strand here is written upside down. See how they have those P's written upside down? Personally, I think they should have the A written upside down. That should be like that. The G should be like that. The T should be like that. Oh my God, drawing upside down G's is hard. All right, so stuff like that. But this is upside down compared to the other. So we have a word for this where one strand looks upside down compared to another. And that word, I guess I have to lose that. That word is anti-parallel. We need a different color. All right, so DNA. The sugar is deoxyribose. The bases are G's, A's, T's, and C's. It is made of 2 strands, and the 2 strands are anti-parallel. Anti-parallel is the word that we use to say, oh, one strand is upside down from the other. Across from one 5' end, there's a 3' end. Got that. Again, I know you guys have seen this stuff before, but I just feel compelled to talk more about it a little bit. Anti-parallel, and they are complementary. I think I spelled complementary right because that's supposed to be an E. If that's an I, that's a different word. Complementary with an I is like, Ma, you look fabulous today. Wow, that was a really excellent lecture today, Professor. Thank you so much. That's quite a compliment. No, complementary has a different meaning. It means things fit together in a certain way. And complementary simply means that across from every G on one strand of DNA will be a C on the other. Across from every T on one strand will be an A. And that's how they figured out, that's what took them so darn long to figure out how 2 strands of DNA fit together, because they knew that across from every A was a T. They knew that A paired with T and that C paired with G, but they couldn't figure out how. The geometry just didn't work until Rosalind Franklin figured out, oh, they are anti-parallel. That made, like this G right here, if we can look at that one, right there, and I'm going to change the color. This geometry allowed this G to make, see these 3 dots here? These dots here. These represent hydrogen bonds. So the C and the G were able to make hydrogen bonds to each other. And I really shouldn't draw them like lines like that. They're really more, they're dots. Hydrogen bond is what weakly holds 2 water molecules together. It's a very weak magnetic attraction from polar molecules. And hydrogen bonds are not just for water. They occur with other molecules as well. 3 hydrogen bonds form between G's and C's, and 2 hydrogen bonds form between A's and T's. So the 2 strands of DNA are anti-parallel, complementary, and held together by hydrogen bonds. Which can be broken. Hydrogen bonds can be broken with just actually a little bit of heat, and that's one of the ways that PCR works. So like the PCR test for, RT-PCR test for coronavirus actually has to separate, actually forms a piece of DNA from the viral RNA and then makes copies of that DNA by basically breaking apart the hydrogen bonds with heat and going in and making more copies that way. So they're held together with hydrogen bonds. And the hydrogen bonds make it possible to make copies of DNA, actually, and also RNA. So there we go. Okay. Okay, so that is DNA and RNA. That is nucleic acid. We're moving on to another favorite, proteins. Proteins are polymers made of amino acids. But before we get into the structure of the amino acid, I want to talk about the functions of a protein. Proteins are the workhorses of the cell. Meaning they do all kinds of stuff. Holy crap, they do so much. Your cells would not survive without proteins. If your cells couldn't make proteins, you would not live. If bacterial cells can't make proteins, they are screwed. They cannot do anything because proteins do so darn much for the cell. You know what? I do not care if you can tell me the difference between adenine and guanine, cytosine and thymine. I am good with that. You are not going to need to tell me which ones are the purines and which ones are the pyrimidines. I'm going to skip over that. Already talked about nucleic acid structure. Strands antiparallel complementary held together by hydrogen bonds. RNA has ribose instead of deoxyribose. Okay, we're just going through the PowerPoint here and making sure I hit all the points here. Proteins. All right, proteins are hugely important to your cells, to bacterial cells. Have here that about an estimate 600 to 800 different proteins in an E. coli bacterial cell. So many different proteins, so many different functions. The first of which would be that they catalyze chemical reactions that are necessary for a cell's survival. Okay, these are enzymes. You already know what enzymes are. You studied those in another class. They are proteins that catalyze chemical reactions for the cell without being used up or changed in any way themselves. If there is a chemical reaction in the cell that requires an enzyme, that chemical reaction won't happen unless that enzyme is there. So cells need enzyme. Your cells need enzymes. Bacterial cells need enzymes. But that's not all. Not all proteins are enzymes. Some proteins comprise different cell structures. So for example, this is not a very well done PowerPoint here. This down here, flagella are made of protein. That is an example of a cellular structure that is made of protein. A flagella is like a tail that a lot of cells use for locomotion. They use it for movement. E. coli has tons of flagella, and that's one of the ways that it moves through a liquid medium. So bacterial flagella are made of protein. So we've got enzymes, we've got proteins. They help nutrients get in and out of the cell. That would be transport proteins. So how's stuff going to get out? You know, your cell membranes, bacterial cell membranes can't have a gigantic hole in there if something needs to get, if something large needs to get in and out. Proteins can transfer those in and out. All right, proteins are also involved in turning on and off genes. Not all genes are active 24/7. If you think about it, if a gene is a set of instructions for how to make a protein, can you imagine that there are times when you may not need every single protein in your genome? Bacterial cells too. Sometimes they need certain proteins to be made, and sometimes they don't. So let's say that E. coli has a gene for lactase. Okay, lactase would be an enzyme that breaks down lactose. Does that make sense? So it would make a copy of it in RNA and then make the protein, which is lactase. Well, why would E. coli want to make a whole bunch of lactase? Why would it want to make a whole bunch of enzyme that breaks down lactose if there's no lactose around to be digested? So there are proteins that will turn off the lactase gene. So these are regulatory proteins, really. Bacterial cells have them, and we actually learned about genetic regulatory proteins in bacteria before we even learned about them in the human human body too. So proteins do a whole lot of things. Okay. All right, they are polymers, they're subunit, dun dun dun, 2 question marks right there. That's already out of the bag. Their subunits are made out of amino acid. What does an amino acid look like? An amino acid looks like this. Okay, when I see the word amino, I think, aha, I know what an amino is. An amine group is a nitrogen atom with a couple of hydrogens hanging off of it. So there's the amino end of the amino acid right there. Hanging off of it, nitrogen has to make three covalent bonds. There's a central carbon, hydrogen hanging off of it, and the acid end of a carboxy acid is of an amino acid. I said carboxy acid is a carboxy acid group right there. So this whole thing there, that's actually a carboxy acid. It is the acid end of an amino acid right there. Ooh, but what's missing? Check out this carbon in the middle here is only forming 3 covalent bonds. Down here, there's what we call an R group, and that is just a placeholder for anything. I could put anything I want there at all. Well, I could, but in nature there are only 20 different options for an R group. So the simplest thing that I could probably put there would be, let's say, a hydrogen atom right there. This is glycine, the amino acid glycine. If I drew another amino acid, let's see, the amine group. Oh, I shouldn't draw these so big. I can only fit 2 in here now. And here's the carboxy end up, and the R group. Why don't I do one of these like that? Just a little CH3 group right there. That's, I think, that's alanine right there. But I'm telling you this because whatever the R group is, that's what changes about an amino acid. If I change this to something else, it won't be glycine anymore. What makes alanine different from glycine is that shouldn't put a circle there. What makes alanine different from glycine is the R group. They have a different R group. Right there. So let's see if I drew this right. Ah, I did. Look, here's the amine group on the left, a carboxyl group on the right. We've got a central carbon here. We've got our R group. Sometimes they're called side chains. I'll call them R groups. Side chains, R groups, same thing. You change the side chain, it's going to be a different amino acid. So just to show you what the different options are. Okay, so up here on the left, glycine. I don't want to do it that way. Come back. Here we go. Glycine. Okay, so the R group is highlighted in blue, and up here in the upper left-hand corner, if that R group is just an H, bingo, it's glycine. Yay, I got that right. If it's a CH3 group next to it, it's alanine. Yay, got that right. Valine, you have some slightly different stuff. I gotta stop tapping. Every time I tap the screen, it enlarges or shrinks it back down. I'm trying not to, but it's so hard not to tap this with my stylus. Okay, so the ones up here where it says where there's nothing but carbon and hydrogens there, those are hydrophobic amino acids. Those are going to be wherever those amino acids are in a protein. It's not going to want to be around water in the cytoplasm. These amino acids of a protein are going to want to tuck towards the middle and get away from the water. So think of it that way. Now down here in the lower left-hand corner, I'm just going to take a screenshot because I can't stand not to be able to tap the screen. Right here. It says alcoholic ones, they're hydrophilic. They just have these alcohol groups, and the alcohol groups make it a little more water friendly. So for example, this thing over here, phenylalanine, it's actually one of the amino acids present in NutraSweet. It's aromatic. There is nothing but carbon and hydrogen here. It's very hydrophobic, it says. But look, if you put a little alcohol group on it, like tyrosine, that makes it hydrophilic. This thing likes water. It's okay with it. FYI. So you got some hydrophilic and you got some hydrophobic amino acids, and depending on where those are in a protein, that's going to help determine what a protein shape is like. The hydrophobic ones are going to want to tuck into parts of the protein that will be away from water. The hydrophilic ones will want to be on a part of the protein that are actually close to water, whether that's the cytoplasm of the cell or the watery exterior of the cell. Do I have some more here? Oh, yes, I do have some more here. All right. Yeah, so you got some hydrophilic ones. You got some hydrophobic ones. You do not need to memorize the structure of any of these. I don't want you to spend your time doing that. It is no big whoop. Do pay some attention to this one down here in the lower right-hand corner where it says L-proline, prol. Check out how the amine group hooks in and around to the central carbon. That R group is kind of funky, isn't it? And it forms this pentagon, doesn't it? All right, where proline is in a protein, it tends to form a little bend to that protein and helps it curl around a little bit. So that's just kind of an interesting one. Wanted to show that to you. All right, so we've got, I kind of lied to you when I said, oh, I can put anything I want here for the R group. Well, I mean, I could if I were just synthesizing it in the laboratory, but there are only 20 amino acids, 20 naturally occurring amino acids in our universe. Maybe other universes will have some other ones. I'm sure that's quite plausible. So there are only 20 different amino acids, but proteins can be hundreds to hundreds of thousands of amino acids long. So can you see you can have an infinite combination of amino acids? The order and number of amino acids, there are just infinite combinations. And that's the first part of how a protein gets its shape. So protein shape is really important. So shape is determined by, at the primary level, and that's what this means, the number 1 with a little degree sign here, this means primary. At the primary level, protein shape is determined by the order and number of amino acids. I'm abbreviating amino acids with AA, right there. So, you know, you could have, and I cannot draw 1,000 of these things and put them together. I'm going to draw them like little beads, right? So depending on the order and number of amino acids, maybe you'll get that little proline thing in there and it'll form a little bend in the amino acid. So certain orders of amino acids, and I'm getting tired of these beads too, folks. I cannot draw these beads. I'm just going to draw these. I'll draw this as a line now. But you can get something that looks like a slinky. That would be at the secondary level. And this slinky, we don't call it a slinky. I would personally love to call it a slinky, but it's really called an alpha helix. So at the secondary level, you can get an alpha helix. Alpha, this is the Greek letter, alpha from the Greek alphabet. We use it a lot in science. It looks like this slinky thing, which is, or an old-fashioned telephone cord. I haven't seen one of those in a million years. Or sometimes, depending on the order and number of amino acids, you can get something that's really flat and folded. And I'm kind of drawing this to look like ribbon candy that you get on the Cape or something like that. This is called a beta-pleated sheet. So at the secondary level, you can get an alpha helix or a beta-pleated sheet. Kind of like, I don't know, a folded paper fan is probably a better analogy than ribbon candy. There we go. Alrighty, but let's say depending on the order and number of amino acids, you can get an alpha helix and maybe 2 alpha helices and maybe a beta pleated sheet and then another one, and then it folds over on itself like that, and it's just this hot mess of a blob. A protein can look like a blob, but I won't call it a blob. Somebody's like, What if blob was one of us? Just a glob. This is actually what we call a glob or a globular protein. It has a very 3D kind of look to it. This would be the third level or the tertiary level of protein shape. A protein can be globular. But not all proteins get all folded onto themselves and look like, a ball of snot. Sometimes they'll look very long and lengthy, like that. Like maybe you can have a really stretched out alpha helix, kind of like that. This is a fibrous protein. And so proteins at the tertiary level can be globular or fibrous. Muscle proteins and motor proteins are going to be shaped like this, very, very long and fibrous, because they're the ones that can stretch and contract, right? Enzymes would be globular, and they'll have their active site here, for example. A lot of proteins are done at this level, but some of them will actually have a tertiary, I already said that, quaternary level, fourth level. And I don't know, I just call this a megaprotein. This would be where you have more than one glob together or more than one fiber together. So, like, let's say there are 4 globs that are bound together and form a megaprotein. Hemoglobin does this. So that's an example of a protein with a 4th level, a quaternary level of structure. There is a protein there's an animal protein where I think it's like, is it 3 fibers that are all twisted together, kind of like a rope? And it's called collagen. You've probably heard of collagen. It's prevalent in your skin and your connective tissue, and it's like a rope that holds your cells together. Collagen is like 3 or 4. I can't remember if it's 3 or 4. I think it's just 3 major ones, even though there are like 6 different types of collagen. But these fibers are twisted together to form a thick rope. Collagen has a 4th level, a quaternary level of structure. Yeah, so let's see. We already got the primary structure, secondary, tertiary, and then some will have a quaternary structure down here in the lower right-hand corner of your screen. These look like 4 globular proteins together, and also at the bottom of the screen down here you have 1, 2, those are 3 fibers together, so that would be a 4th level. Nope, not working for me there. All right, why is protein shape so important? Because shape determines your function. If an enzyme loses its shape, it's not going to be able to do its job. If the rope of collagen untwists, it's not going to hold your cells together all that well anymore. And that's actually what happens to connective tissue as you get older. And, the collagen in your skin, the rope untwists a little bit and you get wrinkles. Like I have wrinkles. Alrighty. But we use that idea of being able to mess up the shape of a protein to actually help us destroy bacterial cells. Because if we can destroy bacterial proteins, those bacterial proteins aren't going to be able to do their job anymore, right? So they get misshapen. So here we have, you guys are going to get tired of me taking screenshots, but let's say this is a bacterial enzyme that helps it metabolize sugars or something. If we heat that bacterial cell up to 100 degrees C, if we boil it, right, we are going to totally screw up the shape of that protein, and it's going to open up and it's not going to be able to do its job anymore. Like, let's say this is the active site right in here. Well, over here, this active site is all gone, right? We say that the protein has been denatured, and that means that it has lost its shape. We use heat to preserve food all the time, and what's key to preparing food for human consumption is to destroy any harmful bacterial cells that are on it, like that are on meat. Okay, so we cook meat with heat, and that denatures and destroys bacterial proteins that are on the surface of that meat. Unless we're talking sushi, but if you're into sushi, you know that you got to get sushi grade fish, for example, and you got to eat that really fast. Or there's a meat called carpaccio. There's a raw food called carpaccio, which is basically raw beef. So not sushi, sushi's fish. But that raw beef better be not that long off the cow. Okay, that stuff for sushi and for carpaccio, any meat that is eaten raw, that is prepared to be eaten raw, needs to be of a certain grade. It needs to be exceedingly fresh. So like we talk about sushi grade fish. It can't be sitting around, and wrapped in plastic on a styrofoam piece of thing in the grocery store, Stop and Shop, in the meat department there for days. No, it's got to be much fresher from the boat than that, fresh from the wharf. All right, but we use heat to prepare food because it denatures bacterial proteins. Another way to do that is with pH. Oh, no, we're not moving there yet. Another way to denature proteins is with pH. Okay, if you lower the pH of proteins, so I'm going to just take the screenshot all over again. You can also use pH, like big changes in pH, like an acidic pH, very acidic pH, will denature bacterial proteins as well. So we use acid brines to preserve food as well. We pickle things. Or if you make something called ceviche, it's common in Puerto Rican cuisine. When I lived in Texas, we used to have ceviche on like a white flesh fish like flounder or shrimp, for example. And what you pour a bunch of lime juice, which is very acidic, over the fish, and it cooks that meat. And it inactivates bacterial proteins that are on the shrimp, that are on the fish, and it changes them so that they're denatured, can't do their job anymore, and that really kills the bacteria. So that's one of the ways that we use denaturing bacterial proteins to preserve food for human consumption. You can also do that with salt to dehydrate things, because a lot of bacterial proteins have water attached to it and they'll lose their shape there too. So yes, we actually do use salting and dehydration as well. Okay, so that's my spiel on proteins. So that leaves us with lipids. Just stay right here. Okay, lipids are basically defined as an organic. Lipids include fats. So not all lipids are fats. So this PowerPoint I should rework this a little bit. But a lipid is basically just an organic substance that is insoluble in water, but is soluble in other organic solvents and fats. Right, so it's not it's a very loose definition. And what's weird about lipids is that, okay, just like we're like, okay, polysaccharides are made up of monosaccharides, and nucleic acids are made up of nucleotides, and proteins are a whole bunch of amino acids strung together. Lipids really aren't like that. They're not quite polymers. They're not made of these repeating subunits at all. Fats have a certain structure to them, but not all lipids are fats. So let's talk about their function. Lipids are extremely important in cell membranes. In fact, cell membranes are made up of lipids. All right. Okay, so they are not always composed of similar subunits. Okay, again, I should have another bullet point right here that says, okay, yes, lipids can be long-term fat storage, long-term energy storage as well, but we're actually going to focus on their importance in cell membranes. All right, so they're not always composed of similar subunits. There are two basic kinds of lipids, simple and compound. And instead of going back to notability, I'll just take a note here and say that simple lipids just have carbon, hydrogen, and oxygen, and that's it, nothing else. Compound lipids have carbon, hydrogen, oxygen, plus something else. A mystery element, whatever. So that's the only difference between simple and compound. Now, before you think that simple lipids just have a simple structure, think again. I'm going ahead a couple of things here. All right, check out cholesterol down here. So the second one, B, and I'm motioning with my pen all the way around it. Cholesterol is a simple lipid. I don't see anything there but carbon, hydrogen, and one little oxygen way down here in the left-hand corner in red. That's it. But this is not a simple structure. Okay, it's a hot mess. You should see how this thing is actually synthesized by squalene in the cell. But cholesterol, which is actually a part of mammalian cell membranes, that would be a simple lipid because it only has carbon, hydrogen, and oxygen. But let's go back to simple lipids and specifically the fats. Fats are simple lipids. They only have carbon, hydrogen, and oxygen, and they're composed of what we call fatty acids and a glycerol molecule. And for this, I know I got a picture of it here for you, but I feel like drawing this just because I can. Let's see. Let's go with navy blue here. All right, I'm going to draw a glycerol molecule. Glycerol molecule. Glycerol is a 3-carbon molecule. Each carbon makes 4 covalent bonds here. Most of them just have hydrogens hanging off of them, but they've got these alcohol groups hanging off of them right there. There you go. There's a glycerol molecule, plain and simple. A fatty acid, and I'm going to draw it this way. The acid part of a fatty acid is a carboxyl group. This makes it acidic right there. And then it's got an R. R group, that's just a placeholder for whatever the heck I feel like drawing on it, but really what it stands for in a fatty acid is just a long carbon chain. Carbon hydrogen chain, nothing else on there. I'm not drawing all these hydrogens here, good Lord, forget all that, blah, not doing it. All right, so to make a fat, we need to join a fatty acid to a glycerol molecule. An enzyme comes along and is like, oh my gosh, we need to get this OH group together with this hydrogen here. It yanks out a water molecule, and behold, we've got a fatty acid, or this would actually be what we call a monoglyceride. A glyceride refers to a fat, right? And a mono means that there's only one fatty acid attached to it. Anybody want to make any bets as to how to make a diglyceride? We add a second fatty acid to it. Okay, and I'll just draw a long carbon chain. I'm not putting all these hydrogens on here. Forget that. I don't have time for that, but yes. We'll join those two, pull them out as a water molecule, and bingo, now we've got a diglyceride. Can you see that if I put another one on here, we could call it a triglyceride? By the way, when people have blood work done, they'll have their cholesterol levels checked, their HDLs, their LDLs, and they'll have maybe their blood glyceride levels done. This is what it refers to. All right, let's see if I drew this right. Oh, I did. I just drew it backwards from the way this McGraw-Hill has it. They've just put the glycerol here on the left, on the right, and the 3 fatty acids over there. So a dehydration reaction will join these fatty acids. Okay, now they left an R group here because they're lame. They didn't feel like drawing their long carbon hydrogen chain. No, because I want to show you what some examples are. And I think it's oleic acid that's next here. No. Oh, yeah, that is oleic acid. Okay, so both of these, we could count them up. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Oh, darn it. It's because my hand touched this. I'm just going to do the screenshot again. All right, these both have the same number of carbons. I'm not going to count them up for you. Go ahead, count it up. But, how are these different? How are these the same to each other? They're 2 different things because one of them has a double bond right here. And do you see how that double bond put a bend in the fatty acid? Yeah. Now, which one of these 2, the saturated fatty acid or the unsaturated fatty acid, has more hydrogens? Which one has more hydrogens? If you said the saturated fatty acid, you would be correct. Has more hydrogens. We say it is saturated with hydrogens. This one has fewer hydrogens due to the carbon-to-carbon double bond. And that carbon-to-carbon double bond is planar like that. It actually is like this. So it puts a bend in things. If there's a carbon here and a carbon there, you got this bend there. So anywho, which one of these is going to take up more space on a fat molecule? This one's going to take up more space. Takes up more space. And because it takes up more space and you can't pack as many of them into a smaller area, these are going to, the unsaturated fats will be liquid at room temp. Think about it. These ones, okay, in a solid, remember how we talked about in a solid, the atoms are really, really close to each other. And as they become a liquid, if you heat them up, the molecules get farther apart. Unsaturated fats, these are going to be farther apart because you can't just pack them in as much. Whereas these saturated fatty acids, look how straight they are like that. You could pack a gazillion of those in really tightly. They will be solid at room temp. All right, so things like butter, bacon fat, everything that tastes good would probably be saturated fatty acid. Unsaturated fatty acids would be olive oil, for example, or corn oil, stuff like that. Oils will be. Before anybody goes and tells me, well, what about coconut oil? Because when it's cold in wintertime, my jar of coconut oil is solid, but when it's summertime, it's liquid. And that's because coconut oil is made up of both saturated fats and unsaturated fats. It has both in them, combination. So it's going to be a little in between. Okay. Oh, incidentally, I'm going to take another screenshot of this. This here, have you ever heard of the food industry? The food industry will be like, oh man, these unsaturated fatty acids, these oils are so much cheaper, but they're liquid at room temperature. So if I put them into my Snickers bar, people are going to bite into their Snickers bar and they're going to get all this liquid oil dripping down their chin. They're not going to like that. Wouldn't it be cool if we could break up this double bond here and turn it into a saturated fatty acid? Because then it'll be solid and the caramel is just so much more yummy that way. The Snickers bar is going to be more yummy that way too. So what they do is they undergo a partial hydrogenation process. And if you look in a lot of fast foods, a lot of highly processed foods, it will say partially hydrogenated oils. That's what we're talking about. They will literally take unsaturated fatty acid, unsaturated fats with unsaturated fatty acids on them, and break up some of those double bonds to form saturated fatty acids. But the problem is you can also get what's called trans fats that way. Trans fats basically straighten out this bend in an unsaturated fatty acid as well, and they actually increase bad cholesterol in your blood, so we're supposed to avoid those. So if you look at some processed foods, and I don't know what Halloween is actually going to look like. I already saw the horrible news that they will not be making Peeps this year. I mean, I can't blame them, but we will have no Peeps this year. But my guess is that Peeps are all sugar, so probably not. But I'm betting Snickers bars and stuff like that will have some partially hydrogenated oils in them by the partial hydrogenation process. All right, so those are fats right there. Now getting back to my drawing, my fabulous drawing here. I'm sure I'm dazzling all of you with my mad art skills. Okay, we didn't do the triglyceride here. I've got a diglyceride going on. For a compound fat, for a compound lipid, the bomb for a compound lipid, if we look back here again, a compound lipid has, no, I didn't write that down. That was just on the PowerPoint. A compound lipid has carbon, hydrogen, oxygen, plus something else. The bomb for the compound lipid, this something else, is phosphorus. And that is called the phospholipid, which is the molecule of the cell membrane, whether it's your cell membrane. Don't do that. Come on, Notability. Work with me here. Whether it's your cell membrane or a bacterial cell membrane, they're made of phospholipids. A phospholipid is just a diglyceride, only instead of putting a third fatty acid here, you put a phosphate group. So a phosphate group gets attached here by a dehydration reaction. But a phosphate group is very hydrophilic. It's got like a -3 charge if it's in solution. Whereas all this stuff down here is very hydrophobic. There's nothing there but hydrogen and carbon. So this is hydrophobic. Whereas this is hydrophilic. This molecule has got a problem. They're like two family members that just can't stand each other, the oil and water, right? The phosphate end is going to love hanging out in water, whereas these fatty acids down here are going to hate hanging out with water. All right, so we got to draw that. I can't be drawing this like this. No, let's do a color we haven't done for a while. Okay, so I'm going to draw this here like this, an oval. And this is going to be like the hydrophilic head. This is the hydrophilic head. And these long fatty acid chains here, I'm just going to draw like two little wavy lines like that. And these are the hydrophobic tails. You take a gazillion of these things called the phospholipid, a gazillion of them float into place and they form the cell membrane. Okay, let's catch up and see how far we are. Oh, there we are. All right, so we've got a nice picture of the phospholipid bilayer. Right, I know you already learned about this in Bio 107 or some other biology course, but just briefly, the only way, like let's say here's the watery interior of the cell. There's water in the cytoplasm, and those polar heads, those hydrophilic heads of the phospholipids love the water, so they're going to point towards the water, whereas those hydrophobic tails will point away from that watery interior of the cell. But we got a problem. The exterior of the cell is very watery too. So another layer has to form with the polar hydrophilic heads facing the watery exterior of the cell and those hydrophobic tails pointing towards that inner layer. So it is a phospholipid bilayer. There we go. It's very fluid. It's not rigid. It's got protein stuck in it. That's what this blue thing is here, and this brown thing is here. Brown and blue right there. Yeah. And so this region here, if you can see where I'm drawing the yellow, all this area in here, oh, I love being able to draw on this. See, here's one thing I could not draw right on the screen if we were in person, but I can draw here. But this region, this is a very hydrophobic region. So like this part of this blue protein here, right there, that's, here we go. This part of the blue protein here is going to have hydrophobic amino acids that are a part of it, whereas the ones that are down here facing the watery interior of the cell, those are going to be hydrophilic. Or at least that's a very, very good guess. It's a reasonable expectation. Alrighty, so the phospholipid, it's what makes the cell membrane. Alrighty, so that is enough for today. I know that this microscope is a part of this PowerPoint, but that will be the topic of chapter 3. We're going to be getting into microscopy and the parts of the bacterial cell and how to visualize bacterial cells, and we'll do that next time. So take it easy, folks. Bye-bye.